Good morning to everyone. I myself, Dr. Frank. I welcome you all to the third and the last day of the BMSCON 2022. Today, we will be starting the scientific session for T-cell vaccine and cancer therapy by our guest speaker, Dr. Manas Mandal. I'm here to welcome our chairpersons for this session. I'm very proud to introduce our most lovable and amicable Professor Dr. Vishnubhat sir, who is the Director Medical Research of AVMC. I think everyone around the globe, sir is well renowned, but still I'm very proud to introduce him. His prior experience as Professor and Head of Neonatology, JIPMA. He was the Dean Research and Director of JIPMA. Sir has a very good training of undergraduates and postgraduates in pediatrics, and he was a faculty at National Teachers Training Center, training of postdoctoral DM, that is in neonatology. And Sir has more research papers of more of 380, and he's an author as his chapters in 68 books. Sir is also an associate editor in Indian Journal of Pediatrics and reviewer. Sir has achieved a National Neonatology Forum Fellowship Award, a Lifetime Achievement Award, Lifetime Research Achievement Award during CAMR Conference of AVMC 2022. Sir has a very high field of interest in medical education, perinatal asphyxia, neonatal sepsis, and congenital malformations. Thank you, sir. Now I would ask Dr. Hema to introduce the next chairperson.
Good morning. It's a great honor to introduce the second chairperson of the session, Dr. Vivek Kumar Sharma. The sir is a very well-known personality in the field of physiology. I think no one need not have an introduction about the sir. Still, I want to say that Doctor is presently working as a Dean, HOD in the physiology, the AIMS Rajkot. Sir has completed his MBBS from the University College of Mumbai, College of Medical Science in Delhi, MD Medical Physiology in the Delhi, and sir has got the 16 years of teaching and research experience. Sir received the Professor R.C. Shukla Oration Award and the Gold Medal in Alternative Medicine and Health Excellence Awards, the speaker and resource person in nearly 90 international and national conferences and CMEs. He is a member of board studies of the various universities and the colleges. Sir has published chapters in books, Encyclopedia of Animal Cognitions and Behavior by the Springstuff Publications. Sir has published 50 peer-reviewed articles, editor and reviewer of various high-impact journals. The field of interest is the mind-body medicine, psychoneuroimmunology, and exercise physiology. Welcome you, sir. Now I request chairperson to introduce the great guest speaker of the day, Dr. Manas Mandal. Over to you, sir.
Shall I introduce?
Morning,
Yes.
sir. Please carry on, sir.
Yes, sir.
Please you introduce.
So, good morning to Dr. Manas Mandal and Vivek Sharma, my friend from JIPMA, and all the delegates who have joined and the organizers. It is very nice to go through the CV of Dr. Manas Mandal. He is a Fulbright scholar and professor at the Roseman University College of Pharmacy. He, of course, born in India and then migrated to USA. And he has done his BSc in physiology, MSc in physiology. Then he did PhD from Jadavpur University, probably immunology or physiology, I'm not very sure. But he's an adjunct faculty at JSS Academy of Higher Medical Education and Research, Mysore. He is a Fulbright specialist appointed by the US Department of State's Bureau of Education and Cultural Affairs. And he has been training people all over the world, maybe. And he was in the Birla Institute of Technology for training the students there. And he has got several awards and published papers and books. And he has moved on to immunology from probably physiology. Of course, we can change our faces in this thing, but I think most of the recent works are on immunology and vaccines. And during the COVID also, he has done a lot of work. And I am sure that with this experience in the field of immunology and cancer therapy, we will have a lot of information. Today, he is going to talk on vaccine, T-cell vaccine and probably cancer immunotherapy. And I wish you all the listeners the best of everything from Dr. Manas Mandal. So, sir, you can start your talk.
Thank you. Thank you. Thanks, Professor Bhatt. That was a very kind introduction. I appreciate it. Good morning to everyone in India. It's in the evening in US. We are exactly about 11 hours apart. So it's Friday evening here and Saturday morning in India. Yes, I was in India with a Fulbright scholarship to teach a course on molecular immunology to the Bacterial and Goa campus students. At that time, Professor Jatia and Professor Bhattacharya contacted me. And we kept the communication, and that led for my participation to today's event. I'm really happy to be here, and I look forward to future interaction with AVMC and when I travel to India in future. So let me begin my presentation with sharing my screen. Sometimes that can be a little technical challenge. Hopefully, I should be able to. Do you see my screen?
We can see.
You can see the screen, right? Okay, let me put my camera here. Because in my, okay, let's see if I can bring it to the slideshow mode. Is it visible to you, all of you? All right.
Yes, sir. Yes, sir.
Thank you very much. So let's begin a little bit discussing. I know this is a diverse research platform. Many speakers have spoken on different aspects. You are right, Professor Bhatt. I started my career as a bachelor's and master's in human physiology, but at the time of my PhD work at the Indian Institute of Chemical Biology and Calcutta School of Tropical Medicine, I got into vaccine research. I was trying to develop a vaccine against snake venom at that time. Slowly, I got interested in immunology, and following that passion in immunology, I traveled to US with a postdoctoral fellowship with University of Chicago, then worked at University of Michigan, Virginia Tech, and finally stayed in the area of immunology and vaccine research. So today I'm going to talk about some of the earlier research I did with University of Michigan College of Pharmacy, really looking into whether we can generate tumor antigen-specific immune response to develop a T-cell vaccine towards cancer immunotherapy. So this is the overview of my talk today. Let's introduce the role of basic biomedical scientists in various research and drug development area, particularly in cancer immunotherapy. Then I'll talk about immunity and T-cell activation, what it really takes to trigger immune responses and activation of a T lymphocyte population in our system. And we'll discuss a little bit of immune surveillance against cancer, meaning almost all of us are prone to develop cancer in our lifetime. Unfortunately, that is true. Why majority of us does not develop cancer? Because there is an immune surveillance system works in our system that is primarily mediated by natural killer NK cells, T lymphocytes, particularly cytotoxic T lymphocytes, that regularly monitors our system against the development of tumor. We'll talk about various antigen-specific T-cell responses, what are the different ways it can be generated, particularly utilizing a vaccine delivery platform that I will be talking today, that incorporates a pore-forming bacterial toxin, which is listerolysine O, derived from Listeria monocytogenes, encapsulated in liposomal delivery system to trigger cytotoxic T lymphocyte responses. And with that experimental model, I will show some experimental data, how we could engineer or trigger cytotoxic T lymphocyte responses, in short, CTL responses, that is protective against murine, meaning mice, melanoma model, and lymphocytic choriomeningitis virus model. So this is my favorite quote in the realm of immunology. Without reading the whole quote, this is where it matters to me most. These knew what it was from experience and had no fear for themselves. For the same man was never attacked twice, never at least fatally. That's the description of plague in Athens, the biblical or bubonic plague in 430 BC by Thucydides, great Greek philosopher. What it tells us that if we got exposed to a pathogen, infection or antigen, and if we survive that infection or antigen exposure, then we develop immunity, immune responses. And that was one of the first demonstration, at least in literature, in Western literature, that how immunity and vaccination could work. Of course, in Indian literature and Chinese literature, we have more older ancient references about immunity, immune protection against cancer and infectious diseases. But moving forward, in modern days, first vaccination scientifically was demonstrated by Edward Jenner, a physician in Glasgow, in England, in 1796, when he deliberately exposed a child with cowpox virus, then conferred protection against challenge with virulence smallpox infection. And moving forward, 100 years later, Louis Pasteur, a French scientist, again demonstrated that altered microbial preparations can trigger immunity against virulent pathogens such as rabies and other infections, thus generated the scientific concept of vaccination. So how vaccines really work? It's really a story and cooperation between B and T cells and generating immune response collectively that ultimately triggers cytokine responses, antibody responses, and cytotoxic T cell responses. But as I mentioned here, sometimes it triggers cytotoxic T cell responses, meaning it's a very daunting task to generate a T cell response, which is CD8 positive cytotoxic T lymphocyte responses. Whereas our body readily generates B cell activation mediated antibody responses and T helper cell mediated, which are CD4 T cell mediated cytokine secretion. But nonetheless, taken together collectively, as it's shown in the left-hand side of this diagram, that this pink structure is an antigen presenting cell engulfing the antigen, then presenting the peptides derived out of this antigen. You can imagine this is a viral antigen or a tumor antigen, and this is the peptide epitope. This green bar here is activating the helper T cell, or it can also trigger a cytotoxic T lymphocyte responses. So tumor vaccine can be generated targeting tumor antigens for protection against cancer. And we already have drugs or vaccines towards that. For example, Gardasil, which is very recently approved to be utilized in India to prevent human papillomavirus, HPV, human papillomavirus infection in women, also protects them against cervical cancer. How it happens? We all know that human papillomavirus, actually 4 clades of human papillomaviruses are involved in 90% of the cervical cancer development in women, meaning 4 clades of human papillomavirus has been isolated from the lesions of cervical cancer from women. Then scientists thought that if HPV leads to cervical cancer in some women, then why not we prevent the HPV infection, and thus develop the HPV vaccine, Gardasil. So let's talk about a little bit T-cell vaccine and cancer immunotherapy. But before we do that, let's look into the overall picture of immune responses. When we are exposed to infection, pathogen, or even when cancerous development occurs in our body, 2 types of immune responses are developed. One is on the left-hand side, innate immunity. And on the other hand side, right-hand side, we have adaptive immunity. Today, we will talk about adaptive immune response, developing immunotherapy against cancer, particularly T-cell responses. But nonetheless, through adaptive immunity, which is highly specific, we generate a pool of B and T-cell responses, and that leads to the immunological memory formation. And for future infection protection, disease protection, this pool of memory B and T lymphocytes are the cell types that provides protection in us. But sadly, Vaccination induces primarily adaptive humoral, meaning B-cell mediated antibody responses with very little or almost no T lymphocyte responses, because it is a daunting task to trigger particularly cytotoxic T lymphocyte responses. Moving forward, what I'll show that through a vaccine delivery system, which is a nanoparticle mediated vaccine delivery of tumor antigen and model antigen, we were able to generate cytotoxic T cell responses in animal model utilizing mice as our animal model that conferred protection against mice melanoma model and lymphocytic choriomeningitis virus infection model. So what are the different assays, immunological assays we have used to demonstrate that fact? Cytotoxic T lymphocyte assay, which is a chromium release assay, enzyme-linked immunospot assay, which essentially looks at the cytotoxic T lymphocytes at a molecular level and single cell level, and so is Tetramer binding assay. These are very high specificity and high magnitude assays, meaning they can really identify and isolate a single cell, meaning T lymphocytes that is responding towards tumor antigen specific vaccination or immunization. Also, these assays are equally efficient to determine viral antigen specific T cell activation. Also, we'll show some data on antibody responses that were generated utilizing our vaccine delivery system. Before I delve right into the experimental design and experimental data, let's talk a little bit about the approaches to cancer immunotherapy. There are various approaches over the last about 2 decades. Huge development occurred in the realm of cancer immunotherapy because, as we know, that current therapeutics and medication and interventions such as surgery, cytotoxic drug therapy, such as 5 cocktails of cytotoxic drugs, vincristine, doxorubicin, etc., cyclophosphamide, etc., does not cure cancer, unfortunately, yet. So there were new researches, or there were some fundamental paradigm shift occurred about 2 to 3 decades back that made our attention focused on whether can we generate a immunity or immune responses against tumor antigen, because naturally we fight tumor development through cytotoxic T lymphocyte activation or natural killer cell activation in our body. So that led to the new area of research of cancer immunotherapy. And today there are many, many avenues of cancer immunotherapy, such as cancer vaccine, that I will talk about my research. Adoptive cell therapy, meaning white blood cells, such as dendritic cells, isolated from a cancer patient, can be pulsed in the lab setup with tumor antigens. Many of the tumor antigens, particularly melanoma and breast cancer, antigenic structures are all known and sequenced. We can pulse those dendritic cells, which are fantastic antigen presenting cells, and expand them in the lab setup ex vivo, and then put them in vivo into the patient's body with the hope that they will educate T cell populations, particularly CTL population in the cancer patient's body, and activate them to find and kill cancerous growth in the patient's body. That's adoptive cell therapy. Very recent development in this area is immune checkpoint inhibitor drugs, PD-1 targeting, programmed DEF1 receptor and ligand specific drugs such as Keytruda, nivolumab, which won the Nobel Prize in Medicine and Physiology in 2019 by Jim Allison's over 40 years of research at MD Anderson Cancer Research, and various monoclonal antibody drugs that are also in the market, such as rituximab, that are used for last 20 years to treat non-Hodgkin's lymphoma patient, and various oncolytic virus therapy and immune system modulation can be done with small molecules. So this is kind of an overall view of how the various approaches of triggering tumor antigen specific cytotoxic T lymphocyte activation. And here is the T cell receptor that is being presented with tumor antigen, this red circle here, via MHC class 1 molecule that will be expressed on a tumor cell that will act as a tumor antigen presenting cell And when that happens in our body, or artificially we can engineer that episode through cancer vaccine, then we can trigger a cytotoxic T lymphocyte response. And after that, this CTL will release cytotoxic chemicals or granules such as granzymes and perforins that will trigger hole or punch hole into the tumor cells. Or this, you can imagine this is a virus-infected cells. Same way, virus-infected cells will be killed through cytotoxic T lymphocyte activation. And various other co-stimulatory molecules such as PD-1, programmed death-1, and its counterpart ligand expressed on the antigen-presenting cells. We have monoclonal antibody drugs such as Keytruda, nivolumab. That's very first-line therapy these days for head and neck cancer, small non-small cell lung cancer, etc. And anti-CTLS4, cytotoxic T lymphocyte associated antigens, Pore-specific monoclonal antibody drugs that are used in conjunction with PD-1 targeting monoclonal antibody drug. These are all frontline therapeutics these days to fight cancer. And therefore, although I will talk about cancer vaccine approaches, what we did as our part as a biomedical researcher at the University of Michigan College of Pharmacy several years back, moving forward, various cancer vaccines such as Gardasil and cancer treating vaccines such as Cytosolic that treats prostate cancer, or very recently, chimeric antigen receptor T-cell therapy such as Chimera are already in the market, either individual personal therapy or as a mass therapy such as Gardasil, such as Cytosolic T, etc. And these are the immune checkpoint inhibitor drugs that also fights cancer development by keeping the T-cell activation prolonged through blockade of program death-1 and program death-1 ligand or CTLS4. These are all inhibitory molecules that naturally expresses on the surface of a cytotoxic T lymphocyte. Once our body determines that it has completed its job, we do not want it to be keeping activated anymore. We need to shut it down. But that may be good to fight cancer, not to fight virus infection in our body, but that is not good for treating a cancer patient or fighting cancer, because there are many cancerous cells in the patient's body and at many different places. Therefore, if we can keep the T cells activated for a prolonged period of time through blocking this program death-1 or program death-1 ligand-1 or cytotoxic T lymphocyte associated 4 inhibitors such as ipilimumab, nivolumab, or atezolizumab, these are monoclonal antibody drugs, that keeps the T cell in activation phase for a prolonged period of time, and hopefully that fights cancer, eliminates cancerous growth in the patient's body. So what we did, we targeted CD8 T cell population, particularly with a cancer vaccine that will trigger MHC, major histocompatibility complex class 1 mediated antigen presentation, targeting, activating cytotoxic T lymphocytes, which are CD8 positive CTLs. Where CD8 positive CTLs actually naturally gets activated to fight virus infection, to fight cancerous growth in our body. So there is other type of T cells in our body, CD4s, which does not play a direct role in fighting cancer. That's why I'm not going through the left-hand side of the panel, but I'll focus our attention to the right-hand side of the panel. It is, as I mentioned before, it is a daunting task to trigger CTL responses because the protein antigens, in this case, the tumor antigen got to be processed cytosolic species in the antigen presenting cell, in this instance, a tumor cell itself. Only then it can be loaded on MHC class 1 molecules like this, and then only a CD8 T cell, this blue cell type, can recognize this peptide antigen, and this peptide antigen is derived from the tumor antigen. Can trigger CD8 T cell. Once that happens, then CD8 T cell releases cytotoxic granules and chemicals. These are simple proteolytic enzymes such as perforin and granzymes. They will punch holes in this cancerous cell, which is acting as a antigen presenting cell that will be ultimately killed. So there were many approaches taken by various scientists over the years, decades, starting from 1988, osmotic rupture of the endosome, meaning endosomal compartment, where usually antigens are located once they are engulfed or we receive a vaccination or infection. We need to rupture them so that antigens are released into the cytosolic space of the infected cells or antigen-presenting cells, and various other cytosolic delivery strategy, such as heat shock protein by Pramod Srivastava at he was at the University, I think somewhere in the Northeast University of Boston, he was working at that time. Then came the pH-sensitive liposome that delivered liposomal encapsulated antigen into the cytosolic space. And following all these leads, what we came up with, could we use Listeria monocytogenes, a bacterial infection derived Listeriolysin, a toxin, inside a pH-sensitive liposome to trigger vaccine antigen-specific robust CTL responses that will be protective in nature. So pore-forming bacterial toxins, it comes from a family of, belongs to a family of cholesterol-dependent cytolysins, CDC family, meaning they love cholesterol. That's a component of our cell membrane. Thus, they readily bind to our cell membrane structure. Who are these members? The notable members are Listeriolysin O, Streptolysin O, and Perfringolysin O. These are various bacterial infections. Many infections do release, upon infection, release various toxins. Some of these toxins are released by these infections respectively. And through the release of these toxins, they are known to punch holes in the infected cells. Here it shows that LLO-mediated rupture of phagosomal membrane that releases the bacteria into the host cell cytosol, and that leads to the replication of the bacteria in an infected macrophage. In Listeria infection, macrophage in our body is the reservoir of the infection. Here you can see the Listeriolysin O being activated or secreted by this encapsulated or endosomal Listeria monocytogenes. This is a vacuolated structure. And upon expression of the Listeriolysin O by this bacterium, Listeria monocytogenes, now this double membrane structure being perforated, then the bacteria comes out into the cytosol, it divides, then it protrudes and releases itself into the next adjoining cell, therefore causing cell-to-cell spread. We thought that why not we utilize this particular pore-forming ability of Listeriolysin O to trigger cytosolic delivery of our vaccine antigen. And these are the details of the Listeriolysin O, which is a 58 kilodalton protein molecule out of Listeria monocytogenes and binds to cholesterol. The three hallmarks of this type of CDC, cholesterol-dependent cytolysins, are cholesterol-loving, oligomerization, meaning they start their pore-forming activity as a monomeric structure, then they form an oligomeric structure and invaginates the lipid bilayer membrane of our cell types and goes into the cytosolic space, and their activity can be lost upon oxidation of these molecules. And just not the bacteria do carry these toxins, but many of our own molecule, our physiological molecules, such as complement-derived membrane attack complex and perforin, these all are capable of punching holes that you see here, a model of MAC membrane attack complex due to complement activation that is known to punch holes on its target cells, and such as what is shown in the panel C. These are the red blood cells, and these black dots on the red blood cells were created through punching of the holes by membrane attack complex formation when complement activation occurred on these red cells. Down below, we see the perforin and granzyme released by a cytotoxic T lymphocyte on its target cells, and these are larger pores. Actually, 10,000 copies of those pores are required to kill even one single infection or a single cancerous cell in our body. Therefore, we need to enhance cytotoxic T lymphocyte responses through a vaccine delivery system to achieve our goal of cancer immunotherapy. Once again, current vaccines generate good B-cell responses, but robust T-cell responses needed, for which we need a cytosolic delivery system. And we brought up with this idea that pore-forming Listeriolysin O can deliver antigens into the antigen presenting cell cytosol, triggering CTL responses to take care of the cancerous growth in a model system. So what we used, we created a nanoparticle vaccine delivery system. Here is a liposomal structure, double lipid membrane. This liposome was developed with phosphatidyl ethylenamine and cholesterol hemisuccinate, PE-CAMs, in 2:1 ratio. And inside this liposomal double membrane, we incorporated a tiny amount of listerolysine O that was generated through recombinant DNA technology expressed in E. coli. We purified listerolysine O, incorporated LLO along with model antigen ovalbumin, and subsequently nucleoprotein from LCMV virus infection, lymphocytic choriomeningitis virus infection, to demonstrate that yes, utilizing a liposomal delivery system that incorporates antigen and bit of listerolysine O, we can trigger a cytotoxic T lymphocyte response. And this is our working hypothesis model that with presence of LLO, vaccine cargo is released into the cytosol and loaded onto MHC class 1 molecules triggering CD8 T cell activation, whereas liposomal vaccine without LLO stays inside the liposome, does not get released into the cytosol, thus does not trigger robust CTL responses. So what are the experimental data in our experimental design? So first, we purified listerolysine O and we purchased ovalbumin as a model antigen encapsulated within the liposome and purified them. And here is the LLO purification, and this purified LLO was encapsulated in the liposomal carrier along with ovalbumin, and those liposomes were purified again through a chromatographic system, size chromatography, and that's what we see out of this gel, that this is the LLO band and this is the ovalbumin band. Then we utilize this liposomal ova or without the LLO containing liposomal ova, our control formulation to pulse or trigger macrophage and dendritic cell activation. These two cell types were generated utilizing primary culture macrophage and dendritic cell from murine, means mouse, bone marrow derived hematopoietic stem cells. It's a really daunting task to generate dendritic cells in the laboratory system, but we got really good help from Ralph Steinman, another Nobel laureate who was working at Rockefeller University at that time. He kindly supplied all the reagents and protocol to generate dendritic cells for these experiments. So what we are seeing here, if we are successful in pulsing dendritic cells and macrophages as antigen-presenting cells with ovalbumin inside a liposomal formulation that contains LLO, then we can bring CD8 T cells. That CD8 T cells can be presented with ovapeptide, and we will ultimately get interleukin-2 release, because for T cell activation, we've got to have interleukin-2 release. This is a classic antigen presentation assay, and here we are showing that in macrophages, with the presence of LLO inside liposome, we generate really high level of antigen presentation, leading to the high level of interleukin-2 release. So this is a surrogate assay that essentially tells us that these antigen-presenting cells, such as dendritic cells and macrophages, have been pulsed with ovalbumin, and they have processed whole ovalbumin, which is a 47 kilodalton molecule, into ovapeptide, such as this synphical, which is an 8 amino acid long structure that has been loaded on MHC class 1 molecules, and that has been able to trigger CD8 positive T cell responses. Dendritic cells in this experiment at least are a little bit less efficient than macrophages, but nonetheless, LLO seems a requirement to trigger efficient IL-2 production, meaning efficient CD8 positive cytotoxic T lymphocyte response. And same data showing in an enlarged version, robust interleukin-2 release shown with LLO, but without LLO, we don't see any IL-2 production either by macrophage or dendritic cells. Subsequently, with this observation, we extended into, that was an in vitro experiment. We wanted to have a proof of concept that yes, LLO triggers ovalbumin peptide to generate CD8 positive T cell responses. Now we wanted to extend that observation into in vivo experimental design. What we did for in vivo experimental model or design is that we wanted to immunize mouse with or without LLO, ovalbumin containing liposome twice, 15 days apart, then subsequently harvested the spleen cells from the immunized animals and did a chromium release assay. And through the chromium release assay, both these antigen presenting assay and chromium release assays are 2 to 3 day assay, meaning once those experiments starts, it takes 2 to 3 days to complete those experiments. And literally they have to be monitored 24 hours. One has to literally live in the lab for that time period. Anyways, when we measure the chromium release in a gamma counter or a scintillation counter, that tells us the killing activity by CD8 positive cytotoxic lymphocytes that have been generated through ovalbumin containing liposomal LLO containing formulation. That's shown as that, yes, that generates a high level of cytotoxicity, meaning the antigen presenting cells or APCs will be pulsed with chromiums, and the chromium will be incorporated into their DNA, and CD8 T cells, over-specific CD8 T cells, when they are capable of killing those antigen presenting cells, the chromium will be released into the supernatant, and we are measuring the supernatant specific chromium. And this is a pretty robust cytotoxic chromium release assay, and 1/3, meaning it maintained the dose ratio here. And soluble OVA did not generate any type of response, whereas liposomal OVA itself, without LLO, been able to generate some degree of cytotoxic T cell responses, but which has been enhanced tremendously, significantly with the inclusion of LLO. So that's the take-home message through these in vivo experiments, that yes, we can generate cytotoxic T lymphocyte responses specific for a model antigen. We wanted to enumerate that observation doing another sophisticated assay known as enzyme-linked immunospot assay. What we are seeing here on the right-hand side circle, these purple dots in this blue circle, is this individual cytotoxic lymphocytes that has been generated in the immunized mice in presence of LLO with ovalbumin immunization inside the liposomal vehicle. And then those spleen cells were harvested and stimulated with same ovalbumin peptides in fecal, and they are generating interferon gamma. And with anti-interferon gamma antibody, we are capturing the spots, meaning these are the numbers of the spleen cells containing CD8 positive T lymphocytes being generated in presence of LLO containing liposomal over-immunization. Whereas cells, means animals that did not receive LLO containing liposomal over-immunization, here they generated really about 1/3 less CD8 positive T cell responses, and soluble OVA number 3 shows very low, almost background level of CD8 T cell activation. So this individual cellular level enumeration corroborates our previous observation that liposomal OVA not only enhances the frequency, but are better killer of antigen presenting cell. And subsequently, I will show you the data. Those antigen presenting cells can be tumor cell types or viral infection mediated, LCM infection mediated, infected virus infected cell type. And the previous data, when we plotted in numbers, this is the differences. About 300 cells per 1 million cells are CD8 positive T cells for ovalbumin and peptide antigen with LLO, but without LLO, we still generate about 1/3 of the numbers. Finally, we went one step ahead. We wanted to visualize that in a flow cytometry, where what is the frequency enhancement. Utilizing a tetramer binding assay, meaning MHC class 1 molecules were loaded with the peptide of ovalbumin in fecal, then that was used to tag or target cytotoxic lymphocytes that are generated due to our vaccine protocols. We see that with LLO, this is nucleoprotein, this is the LCMV monocytogenes specific, antigen specific, nucleoprotein specific peptide frequencies, CTL responses, 3 times at least more without LLO-NP only, and just LLO immunization does not do anything. So for these experiments, we recommendedly express nucleoprotein of lymphocytic choriomeningitis virus in the laboratory, then purified nucleoprotein incorporated within LLO or without LLO inside the liposomal delivery system and immunize the mouse, and following the similar protocol as of before, spleen cell specific cytotoxic T cells being enumerated by this experimental design and protocol. And this Tetramer binding assay and L-spot assays are the top of the sensitive assays still now to determine efficacy of cancer immunotherapy in patient. But while we're doing so, we also wanted to make sure that, are we generating any antibody responses? Yes. All the formulations did generate antibody responses. So take-home message is that although we are focusing on T cell responses, particularly cytotoxic T cell responses through this LLO-liposomal formulation, yet we did not abrogate or diminish antibody responses in the immunized animals. So this is what we have observed so far. LLO-containing liposome delivered antigen efficiently in the cytosolic processing pathway and triggered higher frequencies and stronger cytotoxic lymphocyte responses compared to non-liposomal, means not non-liposomal, non-LLO-liposomal immunized group of animals. With that observation, we hypothesized that can we translate that observation into protection in vivo? 2 models we decided to utilize: lethal melanoma tumor model in mouse and lymphocytic choriomeningitis virus infection and challenge model in mouse. So we immunized mouse over LLO, including—
Sir, we may have
Yes.
to conclude.
Okay, I'm about the time. So utilizing this model, we are showing that our LLO immunized mouse are protected against melanoma containing ovalbumin expressing tumor that is implanted into the mouse. This is the survival date, and this is the tumor progression, and this is the survival of the infected mice in LCMV model. So summary, let's summarize, give me a minute here, that inclusion of liposomal LLO generated tremendous level of CTL activity and frequency that protected mouse against lethal melanoma challenge and protected them against lymphocytic choriomeningitis virus challenge model. These are some of the future experiments ongoing in the lab. And we are particularly interested in looking into CD8-CD4 ratio and targeting toll-like receptor to trigger a robust adaptive immune responses. And these are the conclusions we make. And acknowledgement. I'm grateful for recipient of a Nehru Fulbright Scholarship. I received Upjohn Biotech grant while I was working on EOFM, and these are the individuals as collaborators who helped me doing all those experiments. Thank you. And if there are any question, I will try to answer those questions if time permits.
Can I ask one question? Dr. Vivek, shall I ask one question?
Yes, yeah, please.
I think it is a very, very interesting talk, and a lot of original research has gone through. And my question is,
Yes.
see, now we are looking at like papilloma virus, we have a vaccine because we know that is involved in cervical cancer.
Right.
But other cancer, we do not know whether virus is causing or what something else is causing. So is your suggesting vaccine in general, or is it as a treatment because patient already has got cancer?
Yeah. It's a good observation and a good question. Thank you, Dr. Bhatt, for raising that question. So cancer. So there are various approaches. What I described in the context of human papillomavirus and Gardasil and few other formulations on that line, that's already in use in US and European countries, Western world for at least last 20 years. I have 2 daughters, both of them got Gardasil, 2 doses, 6 months apart, about pubertal age and onwards. And now that recommendation has been moved forward to immunize boys because girls got the HPV infection. It's done for majority of the instances, it remains as a common cell. But upon exposure, they remain as a common cell. Exposure happens through sexual contacts or physical contact from the boys to the girls, usually, and through other ways. So the Gardasil was developed on the premise that if we can control the infection, then we can prevent future development of the cervical cancer in a large number of women. That's one thought process. That's preventative approach. But there are cancer vaccines such as Sipuleucel T, that's the prostate cancer specific. We know the tumor prostate cancer antigenic structures, the peptide antigenic structures for melanoma, prostate cancer, breast cancer, and head and neck cancer and lung cancer. We know the antigenic altered antigenic structures because those are the self-antigens. But at some time point when their growth and expression is aberrant, then we develop tumor in our body, unfortunately. So there is always a little different glycosylation or different alteration in the peptide amino acid sequences in the tumor antigen compared to the normal antigenic structure in one's body. So we have on top of the preventative cancer vaccine, we have a treatment-specific cancer vaccine where scientists are taking the approach that since we know the tumor antigen structures, we can synthesize those peptides in the lab. We can harvest T lymphocytes from a cancer patient. We can harvest dendritic cells from a cancer patient, expand them in the lab setup through a process called leukapheresis, where plasma has been transferred to the patient, and these white blood cells, CD34 positive white blood cells, can be expanded in the lab setup, then pulse those dendritic cells or come up with a CAR T cell, chimeric antigen receptor T cell, to re-engineer their T cell receptor just to bind that specific tumor antigen, whose sequence we know. What we can synthesize in the lab, we can pulse those T cells, we can pulse those dendritic cells, then put them back into the cancer patient, hoping they will fight already developed cancer in the patient population. So these are the two broad ways cancer immunotherapy is developed these days. And of course, there are those immune checkpoint inhibitor drugs such as Keytruda, nivolumab, etc. They are the frontline cancer therapy these days for many cancers.
Thank you, sir. Over to Dr. Vivek.
Sir, Dr. Manas, congratulate you for a wonderful talk. It was really absorbing many things I picked up from this talk myself also, like the importance of the T cell response, which we have talked elaborately about it, and how the recent interventions, how LLO means the bacterial toxins can also be used as a delivery system to activate our own macrophages and dendrites. So my question is, considering like we are going through again the COVID-19 pandemic and all these types of situations where we have new types of the viruses and other types of the things about which we are not aware of are coming up, how do you see the research going on in the field of making it so specific?
Yes,
because we don't even know how these two viruses or many other different types of the viruses are going to react, or the things are there. Can you please elaborate on that?
Yeah, that's a very good observation. Throughout the pandemic, my contribution was I learned as much as an immunologist with my own interest as a vaccine researcher, and developed a course on COVID-19. I'm teaching that course, elective course, for the last 3 years. What I learned through my own self-taught mode, that the greatest achievement is mRNA vaccine. Other vaccine delivery system or vaccine formulation, I should say, such as our Bharat Biotech's killed vaccine formulation, is the age-old vaccine technology that still works miraculously. And that is the easiest way to develop a vaccine for any pathogens, particularly emerging infectious diseases such as COVID-19 or any other bacterial or virus infection. But when the mRNA vaccine that was pioneered by Sarah Gilbert's group at Oxford University, who studied these mRNA sequences of MERS, that impacted the Middle East and Hong Kong, Far East in 2009, 2012. hard group almost came up with a vaccine, but the infection got controlled. There was no necessity and there was no incentive to move forward with the commercial development of those mRNA platform and technology. But that it was understood very well how we can stabilize mRNA molecule. mRNAs are tremendously, notoriously unstable molecule. What the beauty of this group with Moderna and BioNTech, which is itself is a story I can tell some other time. Two immigrants like me from traveled from Egypt to West Germany decades back, their husband-wife team, and one is a molecular biologist and one is a physician. And their dream was to create genetic medicine, gene medicine. They saw the opportunity and they went into work after learning the sequence of spike protein, which binds to the S2 receptors in our body to trigger the infection. So whole idea was that if we can create S protein specific antibody responses and T-cell responses, perhaps we can prevent the infection. So
I think we may have to wind up.
Yeah, scientists who did that now in US, give me one more minute. Yes, my timer was up. That was the child. That mRNA platform has a tremendous opportunity, tremendous prospect. Currently, there is a clinical trial going on in US combining this flu vaccine along with COVID-19 new vaccine that has the sequence of Omicron variant BA.4, BA.5. These are the two latest variants of Omicron of COVID-19. So once we have developed this mRNA platform, we can add the sequences. Beauty of this is that with emerging infection, which we do not know today, but may infect us tomorrow, if we can sequence their genome, their genetic materials, we can add those sequences to come up with a quicker and efficient vaccine delivery system and vaccine formulation for protection. Thank you.
Thank you, sir. Thank you so much for a wonderful talk. And it was really, really very absorbing for all of us. With this, I would like to conclude the session on behalf of Dr. Vishnu Bhatt. Sir has given his excellent, sir has given a lot of time and as well as I would like to say that if any person comes up with any queries and all that, they can still put up the query in the chat box so that we can refer it to Dr. Manas, and with that, sir can also respond back to the people at the later time of the day. So.
Sure. Absolutely.
If Dr. Bhatt, if it is okay, then let me just conclude the session. And thank you very much, Dr. Manas. It has been a great honor and pleasure to be with you for nearly 1 hour or so. Thank you very much. Thank you.
It's my honor, sir. Thank you.
Thank you.
Bye-bye.
Thank you, sir. Thank you, the chairpersons. Thank you, the speakers. Sir, it was a wonderful insight on digitalizing O, and it's a new topic which we learned a lot. And as a token of appreciation and gratitude, we would like you to present with a certificate and a memento, though it's online, sir.
Thank you.
Yes, sir, thank you, sir. We would also like to thank our chairpersons for sharing this session. As a token of appreciation, a small certificate and memento to Dr. Vishnu Bhatt, sir. And also to Vivek Sharma, sir. So, thank you to all. So we'll conclude the session. Now I will hand over the moderating chair to the next person. Thank you all.
Thank you.